Today in this video we are going to discuss regarding nitrogen fixation by rhizobia and blue-green algae. The process of nitrogen fixation in nature. The nitrogen is so abundant in Earth's atmosphere, still it is not available to plants unless and until it is fixed in the soil as nitrates, nitrites and ammonia. Plants can absorb or uptake only these compounds from soil to fulfill their nitrogenous demand. The atmospheric N2 is fixed in the soil by two methods. First, physical method, second one, that is biological method. In physical method, through lightning and other natural or physical processes, nitrogen is fixed. About 10% of atmospheric nitrogen is fixed through this process. While in case of biological method, it is the process of nitrogen fixation in soil through different types of microorganisms such as bacteria, blue-green algae, actinomycetes, mycorrhizal, etc. The nitrogen is very difficult to fix as it requires 225 kilocalorie of energy to break the triple bond, and it is next to impossible to fix it into the soil and make it available to plants. But the bacteria have made it possible through their microbial activities. The bacteria in a legume root nodule fixed atmospheric nitrogen into nitrate and made it available to the plants. This process is known as biological nitrogen fixation. This is the major source of nitrogen in agriculture, and this is also known as symbiotic nitrogen fixation. Now, biological nitrogen fixation through living organisms like bacteria, blue-green algae and mycorrhiza is again divided into two types, that is symbiotic nitrogen fixation and non-symbiotic nitrogen fixation. Now, non-symbiotic nitrogen fixation is done by the organism freely living in the soil. Those are called as diazotrophs. They do not show any kind of association with other host, and they freely living in the soil and fix the nitrogen in soil. So they are called as a non-symbiotic or free living nitrogen fixers. And in case of symbiotic nitrogen fixation, this is the prime important process in life of plant. It takes place in a mutualistic way between soil bacteria, that is rhizobia, and legume or non-legumes. More than 60% of elemental nitrogen is fixed through biological nitrogen activities in plant. It is estimated that a total of 50 to 200 metric ton of nitrogen is fixed biologically in agricultural system. The symbiotic nitrogen fixing bacteria associated with legumes include about 14 genera and 98 species. Symbiotic nitrogen fixation is the most efficient, highly economical and most effective process of adding nitrogen into the soil. And this fixed nitrogen is then taken up by plants through their root system. This is the best example of mutual benefits for both the partners and no one is harmed. The bacteria get food, that is carbon source and shelter, in the root nodule of host plant, and in return they give fixed nitrogen in the form of nitrates or ammonia to the host plant required to fulfill their metabolic demands. So, the nitrogen fixers such as rhizobia, blue-green algae are known as diazotrophs. Now, these diazotrophs are of two types, symbiotic or non-symbiotic, or we can call non-symbiotic fixers as a free living. So, let's start with the symbiotic nitrogen fixation by rhizobia. The symbiotic nitrogen fixing soil bacteria are collectively known as rhizobia, which are aerobic and gram negative. The rhizobia fix nitrogen in the root nodule of various plants belonging to the family Fabaceae, those plants who bear pods. For example, beans, cowpeas, gram, moong, groundnut, susbania, etc. The complete process of symbiotic nitrogen fixation by rhizobia is briefly described in this picture. So, before that, uh, the process of nodule formation is controlled by three factors. First factor is nod factor, second nod gene, and third one bacterial gene. So let's see the complete process of nodule formation in legumes nitrogen fixation by rhizobia in detail. The first step in this is of root root hairs of host plant by rhizobia. This is the first step, and the diagrammatic representation of this is here. This one. This is the first step. Here, that is colonization of root hair of host plant by rhizobia. This is the first and initial stage in the formation of root nodule, which begins with the interaction of rhizobia with legume plants. This is the root hair. These are the bacteria. They react with one another. This is for initiation of symbiotic association. In this process, the host specificity of bacteria is very crucial. Different types of bacteria are present in the surrounding of rhizosphere, soil and host plant. The roots of young host plant secrete a group of chemical attractants like flavonoids and betaines. In response to these chemical signal, only specific type of rhizobia are attracted towards the roots and migrate towards the root hairs to initiate the formation of root nodule with the help of nod factor. The nod factors present on the surface of migrated rhizobia binds to the lectin proteins present on the root hairs. So here is a nod lectin bond get prepared. The adhesion of rhizobia to root hair completes the process of colonization of root hairs by the rhizobia. Now the next step, that is second step, curling of root hairs and the formation of infection thread. This is the second step. Here the curling of root hairs and the formation of infection thread. Look at the diagrammatic representation. This one is the second step. This one is the second step. Here infection thread is formed. Now in this step, the lectin and nod factor interaction includes curling of root hair around rhizobia, and at these regions cell wall of root hairs degrade in response to the nod factor, and immediately the rhizobia enters into the root hair and their invagination in plasma membrane forms a infection thread, which we can see in this diagram. Now third step is branching of infection thread and release of rhizobia. So this is the third step. Here this is the infection thread and there is a branching of infection thread and a release of bacteria here in the cortical cells of the host plant. So in this third step, the infection thread full of dividing rhizobia elongates through root hairs and later on become branched. It reaches into the different cortical cells of root hairs. The rhizobia are now released into the cortical cells either singly or in groups. These bacteria are enclosed by host derived membrane called as peribacterial membrane, PBM. Now these bacteria stop dividing, lose their cell wall and become nitrogen fixing bacteroids, and further the infected cortical cell divide rapidly to form the nodule. It is finally connected with the vascular tissue of the roots for nutrient exchange. And this is the magnified view of rhizobial bacteria and host plants symbiotic association, and which togetherly are responsible for the nitrogen fixation, and this is the final root nodule formation. So this is the overall process of root nodule formation, which is helpful for the nitrogen fixation. Now let's see what is the mechanism of N2 fixation in root nodules. So root nodules are the site for N2 fixation, which contains all necessary biochemicals such as enzyme, nitrogenase, Lig hemoglobin, and energy required in the form of ATPase. So for nitrogen fixation, what we require? First of all, enzymes, nitrogenase enzyme, Lig hemoglobin, and energy. Inside the nodule, the nitrogen fixing enzyme, that is nitrogenase, catalyzes the conversion of atmospheric nitrogen to ammonia. But this enzyme is very sensitive to oxygen. It is damaged or become inactive in presence of O2. Hence, oxygen-free zone is created by Lig hemoglobin, which is scavenger of oxygen. It protects nitrogenase from the oxygen. And in this process, about 16 ATPs, that is energy, is required for fixing one molecule of nitrogen. The equation of nitrogen fixation is given below. First, that is N2 plus 8 electrons plus 10 H plus ions plus 16 ATPs. With the help of Mg++ ion and enzyme nitrogenase, it is converted into 2NH4, that is 2 molecules of ammonia plus H2 plus 16 ADP plus 16 inorganic phosphate, that is PN. So ammonia is the first stable product of nitrogen fixation. Actually, two intermediate compounds are formed during the reaction, but they are highly unstable. So the release of H2 is byproduct of this reaction. Recently, that is in 2009, it was found that this hydrogen released in the process of nitrogen fixation enhance tolerance of legumes to abiotic stress condition like drought, temperature, etc. The rhizobial interaction is also helpful in the process of phytoremediation, through which the legume is protected from the heavy metal stress. The symbiosis between rhizobia and legume is facultative and generally initiated by nitrogen starvation of the legume. It is stated that rhizobial symbiosis contributes at least 70 million tons of nitrogen per year, and plants absorb nitrates or ammonia from the soil. The nitrates are abundant in well aerated, that is oxygenated, and non-acidic soil, while ammonia is predominant in acidic or waterlogged soil. Now this ammonia is assimilated, or how uptake of ammonia is done by plant. This ammonia, that is NH3, produced at the end of biological nitrogen fixation in nodule, is protonated to form ammonia ions, that is NH4+, which are used to synthesize different amino acids. And this process takes place through reductive amination, catalytic amination, and transamination. That's it. In next video we will discuss regarding nitrogen fixation by blue-green algae.